introduction to a dihybrid cross. So this is a cross between two individuals. Anyway, I explained this, but there's no harm in just running through it. So a dihybrid cross, this is a cross between two individuals with two pairs of genes, with each gene having two contrasting alleles. So what you're basically looking at for a dihybrid cross is, remember, for a monohybrid cross, we're just focusing on what? On one character trait, isn't it? So for the monohybrid cross, we're just focusing on one character trait, but or maybe for one gene, right? Maybe is it the length of the stem? Is it the color of the seeds? That's what we're just focusing on. But for a dihybrid cross, you are actually looking at more than one gene. So you find that you're trying, like for this one, this example that we are actually going to be assessing. Okay, so you are looking at two individuals, and then you are assessing two pairs of genes, and those genes, they don't necessarily have to be the same. So with each gene having contrasting alleles, okay, so now these genes are combined together in the same hybrid. Okay, yeah, so remember, we're trying to explain the law of independent assortment. So now, in one such hybrid cross, Gregor Mendel chose seed color. So seeds can either be yellow or green. Then the seed shape can either be what? Round or wrinkled. So those are the two character traits that Mendel was looking at. So the seed color and the seed shape. So what he did was, uh okay, so here we've said with each gene having two contrasting alleles. So you see the definition, right? Two individuals with two pairs of what? Of genes. So you see, you've got two individuals, right? You've got these two parents, and then you've got two pairs of genes, and these pairs of genes, they've got what? They've got contrasting alleles. Okay, so now when you do this crossing, pretty much what you're just trying to do here is to figure out in what ways and what genotype and phenotype eventually are the offspring, okay, of these parents. What are the offspring going to be like? So that's just basically what you are looking at. So you're trying to get, if I can use this word, different combinations that these children can have, okay, or the offspring of this couple can have. So that's an important point to take note of. So, having said that, so now when Gregor Mendel got those plants, or a plant which had round and yellow, round and yellow seeds. Now, here is the thing. How did he know that they were homozygous? We talked about the test cross yesterday, right? So that's how he knew. So now these ones, they were homozygous for all their traits, and you know uh capital R is what shows that it's dominant for a what? For a round shaped seed. Then small letter r is of course recessive. Okay, so that's for a wrinkled, for a wrinkled seed. So it means that the one means that this lowercase r can only produce the phenotype when it's what? When it's homozygous. So what? When it's homozygous recessive. So what this means is that for something to be wrinkled, you know that it has to be homozygous recessive, because the recessive alleles can only produce a phenotype in the homozygous recessive nature. Then again, for green again, which is the seed color, the same thing that I've explained. So when you look at the different combinations, if you were to ask yourself to say, how can these combine? So you look at the first parent on the left, you see that those alleles, they can combine in different ways, but because this is homozygous dominant for both, you see that no matter what ways the gametes can rearrange themselves, it means that a gamete from the first parent on the left no matter how much things can try to move around and play around, the fact that they are both homozygous, it means that the gametes, each and every gamete is going to end up with what? It's going to end up with a capital R and a capital Y, which means that it's all the gametes are going to have alleles which are coding for what? For a round seed and a yellow shaped seed. Oh, sorry, a yellow colored seed, okay? But which is round shaped. Then now when we go on the second parent, just try to check if the people that have to be admitted. Ish, okay, I think I'll just start ignoring. It's really disturbing. Okay, yeah, so yeah, but when we go on the far right, what we are having in the far right is that we've got that arrow and that arrow. Okay, so those are recessive. So meaning the same thing. No matter how you move these around, at the end of the day, it's going to be that each and every garment that's going to come from this second parent is going to be what? They are both going to be recessive because there is no way these things can play around with one another. Those alleles, there is no way they can play around one another to become dominant, right? So that's the point there. So that's why they're just not that they're just going to be one, but they've just written RY capital on the left and RY both lowercase, just to show that the other gametes are going to be the same. So there is no reason to really bringing everything up. So that's really the key thing there. Then now when these gametes, so it means that now when the sexual reproduction takes place, when the yeah, so when now that pollination is happening to form the first hybrid, what this entails is when you look at these genes, you know, oh, sorry, when you look at these alleles, you know that uh, the possible permutation, we know that for the seed shape, it will have to go with the allele for the seed shape, isn't it? And the allele for the seed color is going to go with the allele for the seed color on this other one, right? So meaning whatever combinations that we are going to have, it's going to be capital R, lowercase r, capital Y, and lowercase y. Okay, so this means that all the offspring that are going to be produced from a homozygous round and yellow plant with a homozygous wrinkled and green plant, all their offspring are going to be heterozygous round yellow seeds. So that's what this shows. Now, then, when we go a step further, and then we say now, uh, we now cross the what's this? We now cross the generations, right? So by crossing the generation, we mean and now those offspring which were produced in the F1, the first filial generation, when we now get those and then say, okay, what are we going to do here? We get those and say, let's self-pollinate them, right? So we are getting both things that are what? That are heterozygous, right? For round and yellow seeds. So then, of course, we always split them into gametes, right? So you take maybe this to be the female, that to be the male, male, female, anyway. But we discussed what those signs mean. So now the bone of contention here is you look at for this one. So we know that there's going to be the what? The law of segregation. So meaning these particular alleles can be arranged in four different ways in the gametes. Okay, so for the first part, and some people can even do a crossing for this one. So there are people that cross, like you produce a cross for this, but the easiest way to think about it is say this: the allele for the seed shape will have to go with the allele uh, for the color, isn't it? So it can either be capital R and capital Y. So you write that. or it can either be capital R and lowercase y. So you see that capital R and lowercase y. So then you are done with the possible gametes from R. Then with the capital R or the dominant allele. Then for the recessive lowercase r, for the round shape, you say this lowercase r can go with a capital letter Y. So meaning that this is a gamete that's going to represent having a recessive allele for wrinkled and a dominant allele for yellow being the color of the seed. Then again, we can now have recessive R, and that's recessive for wrinkled, and then recessive for the what, for yellow, right? So those are the four gametes that you're going to have. So it means that the gametes that you're going to have here, they are all going to be what? Those are the potential gametes. Okay, and of course, since it's the same thing on the other side, it's going to be the same. So now what this simply means is, when we start the self-pollination, we have to come up with a way that shows all these potential genotypes. Okay, so that's why it's called a dihybrid cross. So now we have to show what would happen if this capital ry goes with a this, so that's what we are basically doing in the dihybrid cross. And you see that using the usual cross that we are using can be tricky, so that's why here we use the Punnett square. So we discussed what a Punnett square is yesterday. So basically, I know this may look intimidating, but what you are just doing is like we are doing for the Punnett square, right? Just that this is a 4 by 4. But what you do for this one is you write, so you can either choose to say, okay, my column, the column being the one that's vertical, isn't it? So you can say, okay, my column is going to be for the female plant, then or for the female parent, then for let's say for my row, it's going to be for the male. So then you just start doing the same thing that we are doing. So you write your gametes in the column and your gametes in the row. Then after you've done that, then you now again start like the same one we're doing for the Punnett square. So the capital R, capital Y, and that one. So it's just like you are doing the way we are doing the multiplication table, right? So you start with the first row. Rows are the ones that are horizontal. So the first row, you see that's going to be capital R, capital Y, capital R, capital Y. So you just write that. Of course, you are writing the alleles for shape together. So you have so you can't say ry ry. No, you can't do that. So you have to put the alleles for shape together, and the alleles for what? The alleles for the color together. Even if it's recessive and it's what? It's dominant. So you see there, we've written RR again, the same thing. So after you do that, then you've got the potential genotypes. So it means that when we do this crossing, these are how many? 16, right? So meaning uh any product that's going to come from these particular plants can either have a round and a yellow seed, or can either be wrinkled and yellow. But then the beauty about this is that it's going to give us what we call the genotypic ratio, right? So we talked about this. So it's going to give us a genotypic ratio, and that genotypic ratio is very very important, because for someone that's a breeder, let's say someone that works on producing, maybe let's say yellow seeds, you've seen of yellow maize and the white maize. So those people are going to be interested in this data, because then they know, so okay, so the probability of me getting white maize is higher when I use these combinations. So this becomes very very important for people who are in agriculture. Okay, so for those who are in agriculture, so now we look at the genotypic ratio and the phenotypic ratio. So what you're going to do now is you're going now uh to write this. So what you are basically doing here is you're going to say um which ones are having, okay, so like the way it's showing there, so you say which ones are having capital R and capital Y. So capital R and capital Y. Okay, so that's why there they have put R dash then capital Y dash. Simply means that we are just focusing on the capital, or rather the dominant alleles, okay? And when we look at our Punnett square, we are going to see that those are going to be 9. So it does not matter whether it's capital R and capital R, but so long as it's got capital R in it and it's got capital Y, we count all those and you're going to find that you're going to have 9. Okay, so that's your ratio for that. Then for the second part, now you say which one has got, for the color of the seeds, it's got recessive, it's got homozygous recessive, irrespective of the value of R. So in short, what does this tell you? The first part tells you that because it's R dominant and Y dominant, it tells you that the ratio of round and yellow seeds is going to be 9. So you see, then when you now go to, but of course you can find, yeah, so that's that. Then now this one, R dash, then there you've got homozygous recessive for the color. This tells you that these are going to be the seeds which are going to be round, because capital R is for what? Capital R is for uh dominance, right? So you're going to have seeds which are going to be round, but that's homozygous recessive. So what that entails is that these seeds are going to be what color? Green. Okay, so we've done that. Then we now go to the other ratio. Here you see, here now we are saying, you see those, it's homozygous R, right? So homozygous R, we are looking at those seeds which have got what? Which are going to be wrinkled, okay? Because they don't have a dominant allele in them. So these are going to be our seeds which are going to be what? Which are going to be wrinkled. Then we now say we want those seeds which are going to be wrinkled but are going to be yellow in color. So when we check from our Punnett square, you're going to see that we are going to have three. Oh, sorry, yeah. So ratio is going to be 3, but we are going to have R, both lowercase, homozygous recessive. Then that Y, the fact that it's dominant, it means that even if the second allele that comes, even if it's a dominant or it's a recessive one, it doesn't affect, okay, the seed is still going to be yellow. Then for the last ratio, it's about homozygous everything. Of course, we are only going to have one of that, so the ratio is 1, and this is going to be wrinkled and green seeds. So the phenotypic ratio, there you've got it. The phenotype. Phenotype, what's the phenotype? Phenotype is what you are going to see. So you've got a 9 ratio of round and yellow seeds. You've got 3 ratio of round and green. You've got a 3 ratio of wrinkled and yellow. You've got 1 ratio of wrinkled and green. So this is important because they will ask you in your multiple choice questions. So the genotypical ratio is not very important, because what is important for the farmer at the end of the day is to know what their plants are going to look like. But if they want to be sure whether they are homozygous or heterozygous, that's where now they have to do a test cross, which we discussed yesterday. So now we now go to the hybrid test cross. Having mentioned it, it will be great for us to just go right into it. But before we can proceed, are there any questions? Are there any questions? Are there any questions? Okay, okay. So the dihybrid cross. So about the dihybrid cross, really, yeah. So the dihybrid cross is carried out between. So it's carried out between the F1 hybrid and the two gene double recessive, right? So again, this is something that we discussed. So you do it with the F1 hybrid, okay, and then you are going to what to and you are basically crossing that with the double gene recessive. So meaning you're going to cross it with what? With something that is recessive for both genes. And that's why you can see this test cross here. We are actually test crossing it with what? With something that is recessive for both genes. So there you can see the RR, the lowercase RR and the lowercase YY. So that means it's recessive for what? For both genes. So the nine genotypes are present in the right proportions. So the nine genotypes are present in the right proportion. So what this basically is going to give you an idea. So what this entails is let's look at this. So what this entails is that when you do this test cross, right? When you do this test cross, and then you actually find a ratio of you find a ratio, the phenotypic ratio of 1 1 1 1 1 1, then in that case, you know that whatever you are trying to assess, okay, then whatever you are trying to assess is heterozygous for both genes. Okay, but if you find that you do your test cross, and then let's say if all our seeds were yellow, okay, so let's say we will do this test cross here, and then we find that all our seeds are yellow. This simply means that the okay, so I want someone to answer me. If we did this test cross on something, and then we found that all our seeds are yellow after the test cross. What does that tell us about the about the alleles that are coding for yellow? Are they homozygous or is it heterozygous? So they are going to be what? They are going to be, if it's only yellow that we are getting, then that is what? That is homozygous. That's why no trace of green seeds have been shown, because it was homozygous dominant. Now, genetic recombination in the dihybrid cross. So, genetic recombination is observed in the second filial generation of a dihybrid cross. Now, this is very, very important in that so basically, what we are trying to look at is we're trying to compare the things with which we started, like the characteristics or the phenotypes that we started with, right? So we start with our parent plants or pure breeding plants, which are homozygous, dominant for both, and homozygous recessive for both genes, right? Then after that, we had then the first filial generation, which was the F1 hybrids, and these were all round and yellow, but this was a heterozygous for both genes. Now, when the F1 hybrids are crossed among themselves, you will realize that we are going to get certain interesting combinations. We know that having round and yellow and wrinkled and green seeds, those came from the parents, right? But we see that in the second filial generation, we also get round and green and wrinkled and yellow. Now, those were not in the parental types. Does that make sense? So those were not in the parental types. When you check the generation, there is nowhere where that's happening. So that's why you see sometimes my families, all the children were being dark or maybe relatively dark skinned, and then you find that maybe down the line someone becomes light, and people were like, is it really the child, or maybe someone wanted to put, like to give them someone who's not their child. But it's possible. You can have certain combinations which can occur, okay, which can occur. And that's the example that I gave to say, you can have a nose that's shaped like your father, but then have eyes that are like your mother, right? So it's possible that, so you find that how you be looking, the nose you have, and or sometimes you can even have eyes that are intermediate, okay, between your mother and your father, which was has never been in the family tree. So however, the bone of contention is that these new combinations were absent in the parental types. And so because these individuals okay, so these individuals with new combinations are called recombinants, or they could be recombinants or recombinations if you want, but let's go with recombinations. And as a result of crossing over, so we know that crossing over is something that occurs on meiosis, and then it can also be due to independent assortment, okay, in the F1 gamete formation. So we talked about what independent assortment. Actually, that's where the dihybrid cross comes in. Now we talk about the chi-squared test. Others call it the chi-squared test. Anyway, it's the chi-squared test. So what the chi-squared test is, when Mendel was doing his when he was doing these experiments, he was using a lot of plants, right? So all these things that he gave us were simply ratios. Okay, they were simply ratios. So when we try to replicate these experiments, we may get certain deviations, right? But then we don't just want to rule out and say, you know what, this is not correct, this is correct. No, we want to have a logical way of doing things, right? And so, because of that logical way of doing things, we use the chi-squared test to actually assess whether the results that we are looking at from our Mendelian crosses, they are actually, they are actually in agreement with the ratios that Mendelian had found out, or maybe there is a significant deviation, right? So for chemistry, we've been looking at standard deviation, right? So that's why it comes in handy. So now the number of observations used in the chi-squared test must be based on 30 individuals or more in order to get reliable results. So here is the catch. For every experiment, the more your samples, the better. Let's say we say, okay, someone has got, let's say a plant. We say that when we did the crossing, we are finding a phenotypical ratio of 3 being tall and 1 short, right? But if we bring 4 plants, you'll find that all the 4 plants may be short, or all the 4 plants may be tall. Okay, but does that mean that Mendelian was a liar? No, it simply means these ratios, they are easily appreciated when the sample that you are looking at is very, very large. That's why even for Mendel, he was using very, very large samples, right? In that way, you've got a greater probability of being able to see and appreciate the ratios that you're having, right? So that's the thing about it. So having done that, yeah, so that's where the chi-squared test comes in. So you're going to calculate a value, and then there's going to be a table, okay, like we've been doing for the T test, for the F test, and today we are going to be looking about the Q test, right? So for these tests, you do the same. You do your calculations, you get a calculated value, and then you compare it with the value that is tabulated already, right? So what you do, it's pretty much the same. So if the value that you've calculated is greater than the tabulated value or the critical value, the one that has been put in the chi-squared table, it means that the departure of your values from the expected ratio of Mendel is significant. Okay, so it means that there's a real departure from the what? From the expected ratio or from Mendel's, right? But then if you find that your value is actually the one that you've calculated is actually you know, lower than or less than the tabulated value or the critical value in the chi-squared table, it simply means that the ratios that you found are in agreement and their departure from the expected ratio is insignificant. So in either way, or rather in a short way, they are in agreement with Mendel's ratios. Is that clear? Okay, so the formula that we use for the chi-squared test is going to be this one, right? So x squared, then what you're simply doing is you're going, so that symbol that looks like HZ simply means sum over. I'm sure from what we've looked at about standard deviation, we people should be well acquainted with that. So then O is for the observed values. So in short, it simply means the values which you found in your experiment. Then E is for the expected values. So those are the values which you would expect to have if you use Mendel's ratios. Okay, then you get the difference, you square it, then divide it by the expected value. Don't worry, we'll look at an example. So the tabulated value is obtained from the chi-squared statistical tabulated at 5% probability. So again, the 5% probability is important because sometimes to be 10%, and that affects where you're going to be, where you're going to get your values from and which table really you're going to be focusing in. Okay, so please, like I was saying for chemistry yesterday, pay attention to the confidence level and 5% probability. 95% confidence level in a, so 5% probability. Always pay attention to the percent probability because it determines which column you are going to be looking at in the chi-squared table. And then the other thing to take note of is the degree of freedom. Okay, the degrees of freedom. So df is equals to n minus 1. So n is just basically the number of classes or the number of characters, or put in a simpler way, the traits that you are looking at. Okay, the traits that you are trying to analyze. So let's look at an example. So this is an example. Consider the progeny, which is just the offspring, from a cross between two red flowered plants as follows. So you see that those flowers are what? Those flowers are heterozygous, but they are red. So you see the ratio. What's going to be the ratio? So there we've got the that's the, what do you call that? That's the genotypical ratio. You know, so that's 1 to 2 to 1, isn't it? But the phenotypical ratio, it's red and white, which is going to be 3 red and 1 white. So that's the ratio that we are going to use in our experiment. So we know that R, that's capital, it means that that's dominant, right? So it means that all those genotypes which are going to have capital R, meaning their phenotype is going to be red. I hope that makes sense. So now let's now look at an example. So let's take note of what our ratio was. Our ratio was, our genotypical ratio was 1 to 2 to 1. Then, but the one which is really important is the phenotypical ratio because it's what you are seeing from the experiment. So we take note of the phenotypic ratio, which is 3 red to 1 white. So what then you do is, so let's look at the first example. Okay, so don't focus on the second one, let's focus on the first one. So we've done that experiment. Now suppose we observe 152 red and 48 white flowered plants for a sample of 200 progeny f2 plants. Is this result the same as expected? So in short, we've done this experiment and then you are simply asking yourself is, are these values that we've gotten in agreement with the 3 to 1 ratio that Mendel found? Because our sample is 200, that's way above 30, so meaning we can use the chi-squared test. So what you do here, you write a table. Okay, so the first solution is this one, right? So don't look at the table in from anywhere. Start with me. We are starting with the first column. Okay, so the first column is having that hashtag, which is simply for number, right? So number one, two, and the total. Then the character is what phenotypical traits are you looking at? Okay, so what phenotypical traits are you looking at? So we are looking at what? Either the flowers being red or the flowers being white, and then we've got that. Yeah, so really that's really what's important there. Then we now got the observed values. So what are our observed values? So our observed values, and I think you should do a lab on these things. Did you do a lab on this? Because your lab, what do you call like your lab manuals should actually have an experiment on these calculations anyway. Yeah, so you've got observed. So observed is 152 red, 48 48 white, then total there on down it's 200, right? Okay, great. Then what are the expected values from Mendel? So Mendel tells us that from that crossing of heterozygous, the ratio of red to white should be what? 3 to 1. Okay, so how do you find the fraction there? For the red, it's going to be what? You add the ratios and then you put the ratio, the one that you are looking at. So let's say we say 3 to 1. So we say 3 plus 1 is going to be 4. So to find the ratio or the a number of expected uh, red flowers, so we are saying 3 over 4 times the total number of our sample, which is 200 plants. So you say 3 over 4 times 200, then you're going to get, that was the expected what? Expected 150. Then, having done that, then the other thing that you now do is now you want to find out the ratio for the what? For the white plants. But of course you can see if the total is 200, then the remainder has to be for that. But again, of course, let's just do the math. So 3 to 1, so for white is 1, so it's going to be 1 over, so you add these ratios, 3 plus 1, so it's going to be what? 1 over 4 times 200, so you get that 50 there, right? So those 50 and 50 red flowers, sorry, 150, okay, get me, 150 red flowers are the expected number that we should get from Mendel's ratios. Then 50 white flowers, okay, and then the total we see there is 200 for both. So when you do the calculation for the red, you see you're getting 0.03. So you are just basically using that formula. So you subtract 152 minus 150, square the difference over 150, you get that number. You get 48 minus 50, square that over 50, you get the number. Okay, so there when you add those, because remember the formula is saying sum of this formula, right? So you add those, you're getting 0.11. So that's your calculated chi-squared value. So now what then we have to do is we have now to go to that lovely table of ours and see whether this value is less or greater to the tabulated value. So of course, they've told us that we are looking at 95 percentage. Oh, sorry. We are looking at 5% probability, okay? So, what we do here, you see there where it's, what's 5% is what? 5% is 5 over 100, okay? So, 5% is 5 over 100. So, when you divide 100 into 5, that's 0.05. So you go on that 0.05. Then and then how do we find the degree of freedom? We say degree of freedom, we say number of characters. Okay, so the number of characters, the traits that we are looking at. So we are looking at two traits, right? Either white or red flowers, isn't it? So meaning our degree of freedom is n is 2 there minus 1. So meaning our degree of freedom is what? Is 1. Then our probability, we are going for 5%, which is the same as 0.05. So you see, what value are you seeing there? That's 3.8. So is that value greater or less than our calculated value? It's greater than our calculated value. In other words, our calculated value is less than the tabulated value. So what that simply means is that the departure is not real. So the departure is not significant. So it simply means that what yeah, so it simply means that what the ratios that we've observed, they are almost as good as what? As 3 to 1. Because look at that was 140. What? That was what was our number? Yeah, that was 152. And when we were supposed to get 150, that was 48 when we were supposed to get 50. So you see, it's really pretty much, so you see, because our calculated value is less than the tabulated value, therefore the departure is not real, it's not significant, therefore the observed ratio is as good as 3 to 1. That makes sense, right? So yeah, so that means that the departure is not real, it's not significant, therefore the values that we've observed are they're as good as 3 to 1. So I'm just trying to take some water. So, is everyone comfortable with how to do the chi-squared test? Hello? Okay. So, we now look at the second example. So, we go to the second example. So, for the same thing, for the same things that we did, so we look at, so we are doing the crossing between two heterozygous parents. And so we see, but for this one, we observed 165 red and 35 white flowers from 200, uh, you know, from 200 progeny or offspring. Is the result according to the expected ratio? So let's take note of those numbers. Red is 165 and white is 35. So is that a significant departure? So we do the same thing. Okay, let me have gone too high. So yeah, so we look at the table again. Starting from my far left, you see the characters red and white. Then we now go to the observed. That was 165 red flowers, 35 white flowers, total 200. Expected ratio is still the same, and we're still looking at the same number of total offspring, 200. So meaning it's going to still to be 150 and 50 there. Then we do the calculation pretty much the same way. So our calculated chi-squared value is what? Our calculated chi-squared value is 6.0. So what do? We go back to that table and say, is 6.0 greater, because these have got the same percentage probability and the same degree of freedom. But is the value that we've calculated greater than 3.8? Yes, it is greater. So the fact that it is greater, the fact that the calculated value 6.0 is greater than the tabulated value 3.8, therefore the departure is real. And therefore the observed ratio is not as good as 3 to 1. So that's basically what the chi-squared test does for us. So let's now go into a gene. We've talked about a number of things. So now let's go into the gene and then see what we'll be able to do here. So we are remaining with about 21 pages to finish the syllabus. So that's good. So Uh, what's a gene? So a gene is basically a sequence of nucleotides along a DNA molecule which codes for an RNA or polypeptide, right? So of course this is something that we've already talked about. But anyway, so genes which code for polypeptides are divided into two major classes. So you're going to have those genes which are called structural genes. Okay, so you're going to have structural genes which code for functional proteins such as enzymes, hormones, membrane proteins, antibodies. So these are going to be genes which let's say membrane proteins, they form a structure of something, and these are functional proteins, right? So meaning there are proteins that have got a function in the body. Not to say that the others don't have a function, but it's simply put that these are the ones which are actually being formed to do a particular work. Then the other genes are the regulatory genes. Okay, so these ones, yes, of course they are working, but they are not really doing the actual work. They are just controlling the activity. Think of it like this way: a structural gene is the one that like you can look at it as the one that's digging. Okay, let's say people are trying to make a road, right? You see that there will be the supervisor that may even look relatively smart, clean, and then there will be those who have the pick and everything, the shovel. Those are the ones who will be in the you would actually be doing the actual job. Okay, but so, and then that supervisor is equally important because he's the one that is controlling to ensure that those people can actually do the job really really well. So I feel that's an example that can be very very helpful in understanding structure and regulatory genes. So then we now talk about allele, but these are things that we've really talked about anyway. So this is a particular form of a gene occupying a fixed place on a chromosome. So most genes are going to have two alleles, okay, or allelic forms. So the first one is going to be the dominant allele. So in short, this is an allele which masks or hides the expression of another allele of the same gene. So what this simply means is that the dominant allele, it's going to mask or to hide the expression of another. What do we mean? It means that if we are looking at, let's say, height, okay, uh the allele that's dominant, let's say capital T, whether it's homozygous, capital T, or it's heterozygous, where you've got a capital T and a lowercase t, both plants are going to be tall. Okay, so it's masking the expression of another allele of the same gene, and the same gene is very very important because remember, like we saw on the dihybrid crosses, you put the genes for sorry, you put the alleles for a particular gene together, and for that for the other gene together, right? So it only affects the expression of what, of the same gene. So that's an important point to take note of. Then then we now have the codominant genes. Oh, sorry, codominant alleles. So codominant alleles, these are contrasting alleles of the same gene, which are both expressed in the heterozygous. So what's codominant? Codominant, this means that these are genes where even oh sorry, these are alleles, sorry, pardon me, codominant alleles. These are alleles where each allele is going to be seen in the phenotype even when it's heterozygous. Okay, so the best example is the coat color that's in keto. Okay, so you're going to have, let's say, oh, yeah, so it's determined by what? Okay, so let's just break it like this. So the heterozygote is going to exhibit the character of both parents. So let's look at an example of the color code in keto, right? Is the color of the code of the keto, right? Is going to be determined by two alleles. There's one for red and one for white. Now, when homozygous red, which is capital RR, and homozygous white, which is WW capital, are crossed, they are going to produce RW. But because these are codominant alleles, they are both going to be seen. Okay, so both are going to affect the color of the coat. Okay, so the color that they're going to produce is roan. Okay, so they're going to produce roan offspring, so meaning the color will have some red, will have some white, like that, which passes both red and white pairs on their stain. So, or which poses rather. So you're going to have white here, red there. So it's going to give both alleles are going to be expressed even in the heterozygous form. So that's something that's very, very important about the core dominant alleles. So time is not with us, but next time we'll look at incompletely dominant alleles. So we are remaining with 20 pages to finish the syllabus. So most likely by next week we should wrap up with biology syllabus. Otherwise, with what we've discussed, are there any questions?